Enzymes are proteins that serve as catalysts for reactions at physiological conditions, including constant temperature, pressure, osmotic pressure, and pH. Some enzymes require co-enzymes such as metal ions, and small molecules for enzymatic functions. Enzyme activities are tightly regulated. There are several types of enzymes. The first one is called oxidoreductases, catalyze oxidation reduction reactions. The example is pyruvate with a cofactor NADH. The enzyme is called lactate dehydrogenase. The product is lactate and NAD+. The second type is called transferases, catalyze a transfer group from one molecule to another. This is the example. Alanine alpha ketoglutarate. The enzyme is alanine transferase. This is the product is a pyruvate and glutamate. The third type is called hydrolysis, catalyze hydrolysis catalyze hydrolysis cleavage of bonds with water. This example, this is a triacylglycerol water, and the enzyme is called lipase. The product is glycerol and fatty acid. The fourth group is called isomerases, catalyze the conversion of one isomer to another. This is the example. This molecule is called dihydroxyacetone phosphate. They converted to its isomer called glyceroldehyde 3-phosphate. This enzyme is called triose phosphate isomerase. The fifth type is called alliases, catalyze addition of a molecule to a double bond or elimination molecule to give a double bond. This example, this is fumarate plus water to form S-malate. The enzyme is a fumarase. The last type, number 6, the last type is ligases, catalyze joining two molecules into one molecule by forming a covalent bond or joining two ends of a polymeric chain to form a ring. This example, pyruvate, carbon dioxide ATP. The product is oxaloacetate, ADP, and biphosphate. The enzyme is called pyruvate decarboxylase. The last part, let's take a look at how to name an enzyme. In general, a name of enzyme including two parts. The first part identifies the substrate the enzyme works on. The second part identifies the type of reaction catalyzed. Example, succinate dehydrogenase catalyzes the removal of two hydrogen atoms from a substrate succinate. Pyruvate decarboxylase catalyzes the loss of carbon dioxide from a substrate pyruvate. Some enzyme names are derived directly from the substrate by changing the ending to ASE. For example, lactase catalyzes hydrolysis of lactose.